Hi, I'm Sofie Valk, and I will move from structure to function, also following up on some of the things that Casey Paquola has discussed before. So the question is, how can principles of microstructure help us understand function and cognition? And I did some work that follows up on the idea of the structural model, namely that it matters what kind of structure a region has in terms of its layer structure, which goes from allocortex to agranular limbic cortex to disgranular limbic cortex to laminate regions to granular regions. And this tells us something about its connections and also its development, its ontogeny and phylogeny, and that it also helps us understand differences from plasticity to stability, for example. So it says that structure is key, and these different cortical types can help us understand stuff about how the brain is organized and behaves. So a lot of my work is summarized both in structural nomenclature as well as functional nomenclature, which is like a tricky point that I also want to highlight, because every subfield, so to say, in neuroscience prefers sometimes a different terminology for the same kind of regions. For example, if you look at cytoarchitectural classes based on the work of Maedbh et al., you see that the ideotypic parts of the brain here in primary regions more or less overlap with the sensory motor and the visual network in the Yale nomenclature, whereas unimodal areas are like listed here, and they more or less overlap with the dorsal attention network, but also parts of other networks already in the functional organization. And the same holds for heteromotor cortex, which is largely also equivalent to the frontal parietal control network and a default mode network. And then in the end, we also have limbic classes, which are more or less related to the limbic and ventral attention network. But importantly, there's not a one-to-one mapping between, on the one hand side, architectural classes and functional organization. So to study the relationship between microstructure and function, I looked at microstructural profile covariance, as also described by Casey earlier, and resting state functional connectivity. So as you see here, I computed microstructural profile covariance in a human connectome sample, where I probed the intensity of T1, T2 images along 12 layers that goes from the gray matter to the white matter border. And I covaried each parcel's variation as a function of cortical depth with another parcel, and this created such a matrix. And for resting state functional connectivity, I just did the time series of the seed and the target and correlated, resulting in this matrix. Following, to see if regions that have a similar microstructure, namely that they have a positive correlation, also relate to its functional connectivity, I did this edgewise correlation of both metrics and then projected this on the cortex. So what you see is that regions that have a high correlation are primary regions, as you see here, and there's a low or even negative correlation in heteromotor regions, as you also see here in this plot, and paralimbic regions, as you also see here. So this is a summary as a function of cytoarchitectural class, where you see that ideotypic regions have a high correspondence, but unimotor, heteromotor, and particularly paralimbic cortices have a low correspondence in humans. Doing the same approach in macaque monkeys, where we took less layers because their brains are smaller. We also did microstructural profile covariance and resting state functional connectivity, and correlated both metrics, and there's some relationship. And then we did the same, that we looked at it on the cortex using the Markov atlas. And what you see is there are many more positive correlations across the macaque brain. And here's for comparison the human data projected on the macaque surface with help of Xing Su's method to project human data to macaque data. And you see, although there's a positive correlation, there's also a part of the regions that show a high correspondence between microstructural profile covariance and functional connectivity in macaques, which have a low correspondence in humans. And indeed, when you project this alongside your architectural classes, you see that in humans, unimodal and heteromotor regions are uncoupled, including also paralimbic regions. But in macaques, paralimbic regions are still more or less uncoupled or show less correlation, but unimodal and heteromotor regions do not, because they still have a high correspondence in macaques. And if you summarize this along the Yeo networks, you get a similar pattern, showing that there's least correspondence in the frontal attention and limbic networks, as well as default mode network in humans. And in macaques, there's also low correspondence in particularly the limbic network and also default mode network, but a high correspondence in dorsal attention, frontal parietal, as well as sensory motor and visual networks. So there seems to be some difference, particularly in heteromotor regions where it's uncoupled in both humans and macaques, and not in macaques, sorry. Taking a second approach to understand the relationship between microstructure and function, because first we did just an edge level correlation, but that doesn't say us, it tells us something about its organization, but not that much. So for this, we looked also at Messelam's idea about cortical organization, and that you have like, that this organization goes from unimodal to, from ideotypic to unimodal to heteromodal to paralimbic and limbic regions, as you see here in a sort of network that is this heterarchy of organization. And this you can approach relatively nice with a gradient approach. And this has also been shown earlier by Casey Paquola. If you compare the principal gradients of microstructure organization with that of function, you see that there is some overlap, but there are also some notable differences, because functional organization has its apex in the default mode network, whereas microstructure organization has its apex in the limbic networks, as you see here. So we wanted to know if this was also the case in macaques. And yes, for macaques, we also found these two gradients, and comparing, there is some relationship, although it's not so strong, between the organization between macaques and humans, when projected humans to macaques. And again, you see this dissociation between, on the one hand, heteromodal regions and the other hand, limbic regions, whereas the differences between ideotypic and unimodal regions is not that big when you compare this organization of function and structure. And the same holds true in macaques. So you see here this blob going from the limbic network, which is high in structure organization, and this blob from the heteromodal regions that are higher organized in function, sorry. So this is interesting, because this points maybe that these two metrics seem to reflect a different organizational hierarchy, dissociating from the primary regions that are on the other endpoints of the hierarchy in both measures. And you also see the same pattern arising when using it Yeo networks. So the interim summary is that structure and function are coupled in ideotypic and primary regions, and structure and function are decoupled in transmodal regions, including the heteromodal and paralimbic regions. Relative to macaques, heteromodal regions are more uncoupled. And structure and functional organization, they seem to also make a distinction between heteromodal and paralimbic regions. So taking these two ways to describe relationship between structure and function together, and mapping then the site architectural classes upon them and comparing humans and macaques, I created this model where you see that you have like 4 quadrants and 2 uncoupled quadrants in humans, namely here this quadrant that is higher in resting state, functional connectivity, and that one that is higher in microstructural profile covariance. And when you compare this to macaques, you see that this quadrant that is also uncoupled, that has this high default mode frontal parietal or heteromodal network, that's nearly empty in macaques. And the same holds true if you compare it here. You see also that the default mode network largely fills, and also some frontal parietal control network fills this uncoupled organization that is highest in function in humans, but not in macaques. As you see here, it's still empty. So what I did then is I projected task-based functional activations within this framework. And what you see is those regions that are most uncoupled in humans and less so in macaques, they also reflect like language, autobiographical memory, social cognition, episodic memory, which are also functions that are particularly associated with human cognition. So maybe this uncoupling or this different way of associating microstructure to function in humans in these regions, which makes that they're uncoupled, tells us also something about the cognitive functions that are behind it. So also like bringing this further, like how does this relate to function? I also did another study looking at how functional organization changed when doing mental training to have like a causal idea of how an intervention, so a change in these cognitive skills where you learn them, can also alter brain organization. And for this, to map its relation to functional organization, I look particularly at functional integration and segregation. And this is a metric that you can combine either using graph theory metrics, such as path length and clustering coefficient, but you can also make it using gradients, namely combining the first 3 gradients and then taking the average Euclidean distance. You can see that some regions have a high eccentricity, meaning that they are relatively segregated and have a different functional connectivity profile from the rest of the brain. And other regions are relatively integrated, meaning that they're never at the end of the gradient, and they are also like have a similar connectivity profile, like they're more the compromised connectivity profile. So they're more similar to other networks, as you see here. And then we had like a training design where we trained presence model, affect model, and perspective taking. And this was a training of 3 months in 3 blocks with 2 training cohorts and pretest control cohort that didn't undergo any training, but did undergo behavioral and imaging testing, where we imaged structural but also functional tasks and resting state data. And these were both 80 individuals in the training cohort, so 160 individuals, and the resting state retest control cohort was 90 individuals. And presence, that was where we trained attention and interoceptive awareness, whereas in affect training, we trained social affect and social cognition, social affect and emotion, and perspective training was about metacognition, perspective taking on the self and others. So what we found is that when you train presence, you see that these networks in transmodal, like parietal temporal regions, they segregate more, so you become less similar to the rest of the brain, whereas when you train perspective, they get more integrated, meaning that they get more similar to the rest of the brain, which is also like a way that has been shown that the brain starts to behave when you focus and you do something effortful. And you see this in both cohorts, whereas affect didn't show much change in this metric of eccentricity. However, when we looked at the first principal gradient, we find that the affective regions here in the right anterior mid-insula and also SMG, in both trainings, it showed that it became higher up the hierarchy, so closer to the frontal parietal control and the default mode network, possibly is like linking these more saliency regions with a coordination of this eccentricity of frontal parietal control or multiple dimension networks and default mode network. So yeah, what does this implicate? This implicates that the association between structure and function shows an organization in different kinds of relationships between structure and function in the region. And when looking at its mapping of functional tasks, and also like at my plasticity study, you see that each type of training that also resulted in a different kind of change of the brain is embedded in a different way in this framework of structure and function coupling. So with this, I'd like to thank and acknowledge everybody that helped me along the way, and also my lab, which also has some posters that are like more or less related to this work, namely Binwan, Lina Share, and Shema Bayrak. And yeah, if you have questions, let me know on Twitter, send me an email, and yeah, thank you so much for your attention.
Hi, I'm Paquola. Today we're talking about principles of cortical microstructure and how to map them. So there's a long history of neuroscientists being enamored of the arrangement of neurons in the cortex. So I want to take you on a quick journey through that history and make some pit stops along the way to talk about more modern approaches that we can use to map these principles of cortical microstructure. We'll start with descriptions in the vertical direction, so what's happening within the cortex, and then we'll talk about principles that can explain this differentiation across the cortical surface. So when we look at cortical tissue, it's very immediately apparent that there's variations across depth in terms of the number and density and size of cells. And this was the basis for defining cortical layers throughout history. Early on, there was a lot of debate about whether there was 5 or 6 or 7 cortical layers, as well as the nomenclature surrounding them. However, most neuroanatomists have settled on this 6-layer decomposition as a foundation. However, I don't want to linger on this for too long because it becomes very difficult to apply this cortical layer approach in other areas of the cortex, such as the cingulate. So instead, what we can do is use an alternative approach of taking staining intensity profiles. So we can now do this in an automated way using Big Brain. Big Brain is a 3D reconstruction of a sliced and stained postmortem human brain. And it luckily also comes with the surfaces already constructed. So we have this peel and white matter surface. Now we construct a number of equivolumetric surfaces between them. So the equivolumetric constraint means that we modulate the distance between surfaces depending on the curvature of the cortex. And then we take a line from peel to white matter and we sample the staining intensities along that line. So we have depth-wise variations in staining intensity, and the staining intensity for Big Brain in particular reflects the density and size of cells as you move through the cortical depths. So for this profile in particular, we see some quite distinctive features that signify that it does have a laminar structure in this region. So you see a sharp increase in the staining intensity from layer 1 to 2. And you also see this peak around this mid depth, which is around layer 4, where we know there's very densely packed granule cells often. So while this profile is sensitive to laminar features, it's not constrained by them, which is quite important for making this a more generalizable approach across the entire cortex. Also, if you're interested in this, you can find pre-generated profiles for the entirety of Big Brain. So that's 320,000 vertices across the Big Brain surface, all in Big Brain Warp, which is a toolbox we've been developing recently, and it has lots of codes and tutorials on this as well. So taking this a step further, we've translated the approach to microstructure sensitive MRI. So it's the exact same approach, but now the intensities aren't reflecting staining, but a signal from the MRI. It's optimal to use quantitative sequences for this, but T1 over T2 also works quite well. Most important thing is to have higher resolution voxels. So sub-millimeter resolution voxels are best. And also that means that as we get higher and higher resolution imaging sequences, that this technique is just going to become more and more powerful. And if you're interested in an application that we've worked on recently, you can come visit me at my poster. So how can we be sure though that we're measuring architecture with this in vivo method? So here you can see a comparison of imaging-based profiles with the classic works of myeloarchitecture profiles from the FODS. So here we've selected 4 regions where we could find myeloarchitectural pictures in the classic work, and there are very varied areas of the brain. And we compare these profiles. And obviously it's much lower resolution when we're using imaging and we'll not pick up on all the nuances. But what we can do is use a little trick from histology is parameterize these profiles using the central moments. So the central moments are mean, standard deviation, skewness, and kurtosis. The first 2 and those second 2 are highly collinear. So I'll just show the mean and skewness for the purpose of this presentation. But what we find is that across these 4 regions, a similar pattern of changes in the mean and skewness is evident whether we're using staining or imaging. So you see a decrease across these 4 regions in the mean, but an increase in the skewness. However, 4 data points aren't really a rock solid validation, are they? So I'll talk more later about another validation for this approach as well, actually. But using these moments, it also lets us look at differentiation of cortical architecture in vivo in a really simple way, actually. So you see that the mean increases from anterior areas to sensory areas, whereas the skewness of the profiles, this relates to like how flat or imbalanced the profile is. So it increases from sensory areas to limbic areas. So these are distinct patterns of cortical architecture differentiation that we can pick up on with in vivo imaging. So in previous work, we used this to look at changes in adolescence in particular. Here you can see that in the red regions, we're seeing an increase in the mean of the profile during adolescence. You see every part of the profile is increasing. There's a shift of the profile in intensities, whereas in these blue regions there's a decrease in the skewness because just specific surfaces are changing their staining intensity, so it changes the shape of the profile. So really by looking at these depth-wise changes, we can obtain a more comprehensive characterization of individual differences in cortical microstructure. And you've likely already noticed there's extensive regional differentiations in intracortical features. So now let's turn to the principles of regional differentiation. The earliest insights into discriminability of cortical regions was by Theodore Maedbh back in 1885. So he distinguished allocortex, which are the colored regions here, from isocortex, which was everything else. And this distinction was expanded and also, fortunately, this year really well defined in a paper by Garcia Sabezas et al. In terms of 6 different cortical types. So cortical types differ in their fundamental image or construction of cortical architecture. So I've made a little schematic for us here of what specifically cytoarchitectural features change across these types, because numerous things change across these types. So you see the different types on the surface here. And if we look at the schematic, you see that across the types, there's changes in the size of the granule layer, layer 4. Also, the distinguishability and number of layers. So you see there's sub-layering sometime in layer 5 and 6. Other times it's really hard to distinguish any layers anywhere. There's also changes in the position of the largest pyramidal neurons. But that sequence I showed, maybe I'll jump back. So the sequence, it's not just feature-based, that ordering that I'm showing. That's also a topological sequence across the cortical surface. So that was first noticed back in 1947 by Filimonov. So Filimonov was looking at the neural plate in fetuses, and he found that the allocortex here, the hippocampus, part of the allocortex, and the isocortex was separated by periallocortical region, so a transitionary region between allo and isocortex. And Sanides and others showed that this was a common pattern, that there was always a sequence of transitions through types from allo to isocortex. So would that be from out of the hippocampus, there's a sequence, or out of the piriform cortex, there's the same sequence, but it's just in a different lobe. So then we empirically evaluate this relationship between cytoarchitectural variation and the topology within the mesotemporal lobe, because this is an area where we get both the allocortex and isocortex kind of merged up next to each other. So first, though, we did have to overcome a challenge, which is that the hippocampus and isocortex are typically modeled separately in humans. So here you have it in big brain, actually. And we have the purple isocortex labeled. And then we have this really wonderful manual segmentation from Jordan de Cracker, who was the collaborator on this project, in here in the orange colors. So we developed an approach to bridge between these, essentially chopping off that section of the isocortex and making a nifty little algorithm that joined between these surface reconstructions. So then we could have this continuous model of the mesotemporal lobe. And this provides a way to examine the confluence of cortical types in a cohesive way. And this is all in big brain, I should say. So this is a 40 micron slice, and it was only really possible to bridge these reconstructions using this high resolution histological data. So then we also mapped the distance to the intersection between the allo and isocortex. And the geodesic distance, that intersection, is mapped here in this black to yellow color. So then we tested where the geometry of the mesotemporal lobe, specifically the infolding of the isto-allocortical axis, corresponds to the cytoarchitectural gradient. So to do so, we extracted staining intensity profiles that I discussed before from the whole medial temporal lobe. And then we correlated them to get cytoarchitectural similarity between each point, and then employed diffusion map embedding, which is a nonlinear dimensionality reduction technique, to extract this principal axis of cytoarchitectural differentiation. And we compared this to the geometric axis and find a very strong correspondence between them. So that shows that variations in cytoarchitecture are topologically organized along these large-scale gradients. Additionally, we could ask which features specifically characterize this transition from ice to allocortex. We extracted a set of features from the microstructure profile, specifically the staining intensity at each depth, as well as the 4 central movements I mentioned before. And we fed these features into a random forest regression, where we selected the hyperparameters based on a 5-fold grid search. And then we used 70% of the vertices for training and 30% for testing. And the microstructure profile features provided a very high accuracy in predicting the position along the isto-allocortical axis. So here we have a very well-fit model here. And then we could use an iterative leave one out procedure to test which feature was most influential, most important for our model. And we find that the skewness was most important in decreasing or in explaining variance within this model. And that's quite interesting because, as I showed before, the skewness changes from sensory to limbic regions. And this is an extension of that isocortical model into a deeper area of the limbic kind of cortex. So we see that this axis of isoallocortical transition is a continuation of that large scale gradient that we saw across the whole isocortex. And to show this in another way, we tested whether this isoallocortical axis is contiguous with the sensory-fugal gradient that had been proposed previously. So here we have this sensory-fugal gradient, which is the principal axis of cytoarchitectural differentiation in the whole isocortex. So we resolve this in a really similar way as I showed for the mesial temporal lobe project. But the point I wanted to make is that it runs from sensory regions to more limbic regions, and then you can see the axis that we just showed for mesial temporal lobe continues from there. And both are very sensitive to changes in the skewness of the profile. And we could also replicate this with in vivo data, which was very important. So we have this very solid validation for utilizing the imaging-derived profiles, once again, as this principal axis of variation in sensory-fugal direction. However, this sensory-fugal gradient may be the most dominant axis of cytoarchitectural differentiation, but it's not the only one. The sensory-fugal axis is our first eigenvector, but the second eigenvector shows a distinct pattern more from anterior to temporal or lateral regions. And if we combine these, you see a finer resolution differentiation across the cortex. And importantly, this is an overlap of two different cytoarchitectural gradients, which echoes previous work by Bailey and von Bonnen, who used this watercolor approach to show how cytoarchitectural features change in gradients across the cortex and can overlap. So for example, if you take the yellow granule feature with the blue allocortical feature, you see that they overlap to define this green cingulate area. So to sum this up, we have a way to look at depth-wise variations through profiles. It does not assume laminar structure, which is helpful, and also has flexible utility for both histology and in vivo imaging. Then we also have regional variations that are well captured by gradients because cytoarchitectural features are topologically organized, and then their intersection can actually explain a vast array of differentiation within the cortex. I won't touch too much on functional organization because Sofie Valk is going to do so later in the symposium. So to wrap up, these principles have implications for how we model the cortex more broadly. First, it shows that we need to move beyond the notion of uniformity of the cortex, such as the canonical cortical microcircuit, because not all the cortex doesn't even have all these layers, and I think it just disregards the profound regional variability that we do see in cortical architecture. So it's time to diversify the canonical cortical microcircuit. And also it suggests that multidimensional approaches can provide more comprehensive reconstructions of cortical organization. And so with that, I'd just like to say thank you to everyone involved, especially my mentor, Boris Bernhardt at the Mika Lab for M&I. And also, if you're interested in these tools, go check out Big Brain for more histological in vivo integration.
Hello, my name is Maedbh King, and I'm currently a PhD student in Rich Ivory's lab at UC Berkeley. I'm excited today to contribute to this panel by discussing the challenges that come with imaging the human cerebellum, along with the advances that have been made to better understand the functional role of this underappreciated structure. So I spend a lot of my time thinking about the other brain that's missing from this picture. And by other brain, I of course mean the little brain, or the cerebellum. And this is the brain structure that is at worst excluded from the field of view of many fMRI studies, and at best relegated to the supplementary materials. However, as many of you know, although only comprising 10% of the total volume of the brain, it's actually home to approximately 80% of the total number of neurons in the human brain. And this simply makes it too big to ignore. So given how different cerebellar architecture is from cortical architecture, I first want to spend a bit of time discussing cerebellar anatomy before providing an overview of some of the challenges that are associated with imaging the cerebellum. I'll then wrap up by summarizing some of the recent advances that have been made to better understand the functional organization of this structure. So, like the cerebral cortex, the cerebellum is divided into left and right hemispheres. Unlike the cerebral cortex, there's a phylogenetically old portion of the cerebellum known as the vermis, which integrates visual, vestibular, and proprioceptive information. Now, the image that I'm showing you is a post-mortem human cerebellum scanned at 200 micron resolution. And at this resolution, you can clearly make out the beautiful branching structure of the cerebellum, cerebellar white matter. And you can also see the deep cerebellar nuclei embedded within the white matter of the cerebellum. Now, one important difference between the architecture of the cerebellum in contrast to the cerebral cortex is the incredible tight folding of the cerebellum, which you can see clearly illustrated here in the sagittal slicing through the cerebellum. And this tight folding is a very important feature of the cerebellum that introduces unique neuroimaging challenges, which I'll discuss later. So the foldings of the cerebellum are called folia, and they're approximately 1 to 2 millimeters in width. And if we zoom into a folium, we can see that there are 3 distinctive cortical layers, starting with the molecular layer, which is mostly comprised of axons, the Purkinje layer, home to the major computational neuron of the cerebellum, the Purkinje cell, and then the granule layer, where the 50 or so billion granule cells reside. And this 3-layered architecture is repeated across the cerebellar cortex and is a unique feature of the cerebellum. So, like the basal ganglia, we can't talk about the cerebellum without recognizing that this structure operates within the context of a large corticothalamo-pontine circuit. In fact, in terms of a numbers game, the corticocerebellar system is one of the largest neuronal circuits in the human brain. 40 million axons leave the neocortex through the cerebral peduncles, the vast majority of which project the pontine nuclei. From there, this stream of information is projected onto 50 billion granule cells corresponding to an expansion of 1 to 1,000. Granule cells then give rise to a single parallel fiber, and each fiber innervates 15 million Purkinje cells. Purkinje cells then deliver their output via the deep cerebellar nuclei to the thalamus and then back to the neocortex. So interpreting the cerebellum within the context of the cerebral cortex is important if we're going to further our understanding of cerebellar function. Now, while we know a lot about the anatomy of the cerebellum, the advent of functional neuroimaging really allowed us to further our understanding of cerebellar functional organization. So from the first landmark PET study that showed cerebellar activation in response to single word processing, many hundreds of neuroimaging papers have been published showing cerebellar activation in response to attention, response reassignment, higher order rule processing, verbal working memory, language, social cognition, amongst many, many others. And this research has been critically important in shaping how we think about the function of the cerebellum in humans. Now, despite this large body of work, it's important that we recognize the challenges that researchers have faced in imaging the cerebellum. From physiological artifacts to spatial normalization, there are many reasons why researchers have removed this brain region from the field of view in fMRI research. And one important factor to consider are physiological artifacts. So due to its dependence on blood flow and oxygenation, the bold signal is highly sensitive to changes in respiration and cardiac function. And this is especially true for the cerebellum and the brainstem, which are surrounded by a dense bed of vasculature, as you can see here. So as the heart contracts and relaxes during the cardiac cycle, periodic variations in arterial blood pressure are transmitted along the vasculature, resulting in relatively localized motions and deformations of the brain. And this motion clearly affects the cerebellum. But fortunately, physiological signals are easily recorded, and when they are included as nuisance regressors in a GLM, they can account for 35% of the variance around the brainstem and the cerebellum. Now, a second important factor to consider is the bold signal itself, which relative to the cerebral cortex has a very different origin story in the cerebellum. So presented here is a schematic of the circuitry of the cerebellum. The main computational unit is the Purkinje cell here in green. Mossy fibers bring input from the cortex and the spinal cord to the granule cell, which then synapses onto the Purkinje cell via the parallel fiber. And interestingly, work by Thompson and Matheson and colleagues has found that increased spiking activity of Purkinje cells is neither sufficient nor necessary to elicit cerebral blood flow responses in the cerebellar cortex. Now, what that means for the cerebellum is that it's likely that the bold signal is largely reflective of input signals from the cerebral cortex, not Purkinje cell firing. And this is an important detail to consider when we interpret task-evoked or resting state cerebellar activation. So a third factor to consider is anatomical localization, which has long been a challenge for cerebellar researchers. Presented here in this video is a reconstruction of an individual human cerebellum resolved down to the level of individual folia. And this is the same post-mortem cerebellum that I showed you a few slides ago, scanned for many hours at 200 microns. And color here indicates local curvature, with green indicating the crest of a folium. When this reconstructed volume is flattened, it is estimated at 78% of the surface area of the entire human neocortex. However, due to the current constraints of imaging techniques, it's really not feasible to resolve down to the level of individual folia in our research participants. And even when the cerebellum is resolved to the level of groups of folia rather than individual folia, a resolution higher than 200 microns is required for complete unfolding. And furthermore, the typical resolution of functional imaging data is between 2 to 4 millimeters isotropic. Therefore, the activation from adjacent folia is averaged. And you can see here in this atlas from Van Essen that there are two sites of individual finger representations in the cerebellum, one in the anterior lobe and the other down here in the posterior lobe. However, you can see here that these activations, these loci are fractionated and distributed over a number of folds of the surface. And this problem arises from the fact that the level of detail of the anatomical unfolding is simply too high for the resolution of the functional data. So what then can we do? Well, to address these shortcomings, in recent years, a probabilistic atlas has been developed, which is resolved not to the level of the folia or groups of folia, but to the level of lobules. And this development has taken the form of a spatially unbiased infratentorial template, or SUIT for short, presented here on the left in coronal, sagittal, and horizontal views. And each color denotes a different lobule. And more recently, a flat map representation of this atlas has been developed to allow for the visualization of cerebellar gray matter in one concise view. So you can see here, starting from lobules 1 to 4 in the anterior lobe, traversing them through crus 1 and crus 2, all the way down to lobule 10 in the floccular nodular lobe. And this is a really nice way to concisely visualize cerebellar activation in one view. So now you can see, if we visualize these same motor activations on the group map, the one that's unfolded to the lobules, you get these closed clusters in the anterior lobe and in the posterior lobe, compared to the fractionated clusters in the individual atlas here on the left. And furthermore, the deep cerebellar nuclei, which sit in the white matter of the cerebellum, these can also be normalized to SUIT space. However, the difficulty with the deep cerebellar nuclei is in identifying them clearly on a 3T scan. So they're really small structures. They're comprised of only a few hundred voxels, and they're not always visible on scans. Therefore, it's likely that scanning at 7 Tesla would improve segmentation of these structures. Now, fortunately, the challenges that face cerebellar neuroimaging can largely be addressed, and because of recent advances, we have been able to glean many important insights about cerebellar function and organization. So for the last part of this talk, I want to walk through some developments that have been made in mapping the cerebellum using resting state and task-evoked data. So we know mostly from polysynaptic tracing work in non-human primates that communication between the cerebral cortex and the cerebellum is often understood as relying on a series of parallel loops, each connecting a single cortical and cerebellar region in a closed loop circuit. So for example, Strick and many others have shown that if you inject retrograde tracer into area 46 of the prefrontal cortex, that cells in the dentate nucleus are labeled. And what's important here is that these dentate neurons are located in regions that are spatially separate from those labeled after injections into motor areas of the cerebral cortex, indicating a clear motor, non-motor topography. And this labor-intensive work was done in primates, and it's very invasive. However, resting state fMRI over the past decade has provided a great alternative, a great non-invasive alternative for mapping corticocerebellar connectivity. And a lot of this important work has been done by Buckner, Yeo, and colleagues using resting state data, and they have corroborated the seminal anatomical work by demonstrating a one-to-one mapping between corresponding networks of the cortex and the cerebellum. So they generated a map of the cerebellum by regressing each cerebellar voxel against cortical resting state networks. And then they did a winner-take-all approach to assign each voxel of the cerebellum to the cortical network with which it was most correlated. And this is the resulting map that they achieved. And what they showed was that was supporting evidence for connectivity between motor regions of the cortex, so in turquoise and blue here, and the cerebellum, illustrating this known somatotopy of the cerebellum. And what was really interesting, however, was that the intervening regions between these motor representations, so all these regions here in pink and in purple, these were largely mapped to cortical networks associated with non-motor function, so these pink and red networks in the association regions of the cortex. And more recently, work from the same group has also shown that this corticocerebellar connectivity can in fact be identified at the individual level, shown here in 2 example subjects, and that the individual cerebellar topography contains multiple representations of cortical network, similar to what was shown at the group level. So, if functional parcellations of the cerebellum are to eventually be used for translational or clinical purposes, it really is important that this functional organization can be identified in individual cerebella, as shown here. Now, task-evoked fMRI activity has also been leveraged to derive a functional parcellation of the human cerebellum. And what's notable about task-evoked fMRI is that it allows for semantic meaning to be assigned to functional subdivisions by regressing cognitive features against task profiles. And recent work has found that, for example, regions in the anterior lobe of the cerebellum encode left and right hand motor presses, a known somatotopy in the cerebellum. Another region encodes saccades and other features related to visual recognition. A fourth is a motor region that's driven by action observation. But interestingly, beyond these motor regions, the features most descriptive of cerebellar functional organization captured a range of cognitive processes from attention to narrative, verbal fluency, and autobiographical recall. And this nicely illustrates this sort of rich functional organization of the cerebellum. And similar to the resting state work, these task-evoked parcellations can also be reproduced at the individual level. And, while it's largely a practical decision to go to the group, a lot of information is lost by averaging over individual subjects. So one interesting methodological question for future fMRI research and MRI research will be to determine how much data, resting state or task-evoked, is sufficient for deriving functional parcellations at the individual level. And another way that cerebellar researchers think about functional organization is in terms of functional gradients. So in this recent work, diffusion map embedding was applied to resting state data from the Human Connectome project to show that cerebellar functional regions follow a gradual organization which progresses from language and default mode network regions here in red to these motor regions in blue. And then a second gradient was identified that isolated working memory and frontoparietal network areas. And all of this work, be it functional parcellations or functional gradients, contribute to our understanding of cerebellar functional organization, both by itself and within the context of the cerebral cortex. And finally, making these maps and these gradients available is an important step in advancing the field of cerebellar neuroimaging, because they provide a useful guide in designing studies to test specific functional hypotheses, as well as facilitating new ways to interpret cerebellar activation. So here you can see a brain viewer has been created for visualization of task maps and parcellations, and you can scroll through each of these different maps. You can also download them for your own research. And then the functional gradients have also been made available for viewing in an interactive fashion, and they can be accessed here. So overall, despite the challenges that face cerebellar neuroimagers, I really do believe that it's a very worthwhile endeavor to use MRI and fMRI to study the cerebellum. There's a lot to be learned about this mysterious and complex little brain. So with that, I would like to thank my PhD advisor, Richard Ivory at UC Berkeley, my master's advisor, Jorn Dietrichson at Western University, and all the members of the Cognition and Action Laboratory at Berkeley, who've been a great support in my graduate career. Thank you.
Hello, and also welcome from my side to our session, Putting the Fun Back into Functional Neuroimaging, My name is Andreas Horn from Berlin, and I will talk about the basal ganglia in 15 minutes. So it's crazy a lot to talk about. So I'll try to be brief, and I will probably scratch the surface on quite some topics only. So quick recap about the basal ganglia, then we'll dive into MRI imaging of the basal ganglia, and then talk briefly about some applications such as the brain simulation imaging. If you look at the basal ganglia, they are a set of very small structures in the base of the brain. So from outside, you don't see them. That's why maybe sometimes they get neglected more than they should. And they are small, so hard for imaging. Challenge accepted. Let's dive into it. To quickly recap, we conventionally call the chordate, putamen, and nucleus accumbens together as the striatum. It's one of the main input structures of the basal ganglia. Then we have the external and internal pallidum. Again, a very important structure in the basal ganglia. The subthalamic nucleus, very important for clinical applications such as the brain simulation in Parkinson's disease. And then we have the reticular and compact part of the substantia nigra. And together, these are the conventional parts of the basal ganglia. You cannot talk about them without talking about loops. So you have motor, associative, and limbic loops. Sometimes you have also premotor and oculomotor loops. So going from the cortex over the striatum, through the pallidum, then back to the thalamus and back up. And you could also subdivide, let's say, the motor loop into somatotopic loops. For example, I have a leg loop, an arm loop, a face loop, and so on for the motor system. So, you know, these loops are models as always, and they can be helpful, but they are not a final grain of postulation that we can apply. And if you follow the nomenclature and concepts of Larry Swanson, you could even postulate the whole brain into a cortex, striatum, and pallidum region with always these excitation, inhibition, and then disinhibition functions. So Larry Swanson would postulate all other cerebral nuclei into either striatum and pallidum as well. So he uses these terms a bit more generalized. And to show you what that means, these two loops are basically the dorsal and ventral striatum loops that we know of. But these other loops use different structures to have the same function of the striatum and pallidum. So potentially this general idea of that basic circuit could be translated across the whole brain. Then looking more focused again on the basal ganglia, very important are these box and arrow models, especially the one by Albin and DeLong from around 1990 that some of you have surely seen already in med school or somewhere else. Then from the computational side, Gurney, Prescott, and Redgrave created a model that I think is very important as well, which had this notion that the direct pathway is prokinetic and the indirect pathway is antikinetic. So the idea of, you know, one pro movement and one against movement. And this has evolved a lot, but is still useful sometimes. So then these are the structures again. So you have these direct and indirect and also hyperdirect pathways that functionally play a role. And these have been very influential. I must say also that these wiring diagrams are much older than the 1990s. For example, the Vogts created similar drawings in 1922. So as always, science history repeats itself, and we often neglect what has been discussed 50 years ago already. What is the current state of the art? If you ask, I think one of the basal ganglia experts, Heike Bergmann, whom you can see here looking at intraoperative data, and who just wrote a really nice book about the basal ganglia that I must recommend. It's really fascinating, will be out soon. You can look at his most modern version of the box and arrow plots, where you can see the classical basal ganglia main axis integrated into a reinforcement actor-critic model that receives dopamine, but also input from other neurotransmitters and modulators to like shape the basal ganglia activity. But then you have to, when you go to neuroimaging, you have to translate these functional models into anatomical domain, which is not even straightforward. This is my attempt that you can see here. So this is again this main axis of the basal ganglia going through Wilson's pencils, through the striatum, through the pallidum, and then through the internal capsule in form of the Eddinger's comp system to the STN and substantia nigra. And then there's the pallidothalamic outflow fibers that go back up through the thalamus and back to the cortex. right? So if we want to model and image that, we need a good anatomical definition of these structures as well. And one more integrated attempt that I really love and also must recommend by my co-speaker, Mac Shine here, is really a must read about the basal ganglia and cerebellic integration via the thalamus. So in his new concept from just 2020, the thalamus has the ability to integrate information from basal ganglia and cerebellum and modulate the cortical repertoire based on these inputs. So that is a more anatomical and more, directly applicable functional concept that should be explored more. The basal ganglia also form as a dimensionality reduction system, which you can literally see here on this beautiful image by Markus Axa, polarized light imaging, a section of a coronal slice through the brain of a vervet monkey, actually. So these are Wilson's pencils that I talked about going through the putamen, and then you have some rewiring in the external lamina, then they go parallel again in the external pallidum, then some rewiring again, then you have again parallel fibers in the GPI, rewiring, and then parallel fibers in the internal part of the GPI. So you can literally see how cortical information gets compressed to a smaller space, and then it goes through the thalamus back up and gets expanded again, just as in many machine learning applications that we study these days. A problem, though, with imaging is that the resolution is too poor, right? This is what we just saw, and this is our resolution that we usually have in optimal imaging sequences, right? So we don't see things. What can we do to improve that, at least a bit? So on 3 Tesla, you can basically use 2 strategies. One focuses on iron, one on myelin, with 2 example applications here. This is quantitative susceptibility imaging, which is really good for the iron-dense structures such as the SDN and the red nucleus. And you also see the ANSA here and some other structures. And then the myelin structures are really good for the pallidum, but also for the thalamus. So here you can see the lamina dividing the internal and external pallidum, and then the one dividing it from the striatum, from the putamen. And inside the thalamus, the same sequence gives you quite some resolvence of the thalamic nucleus. So there it can be really helpful as well. Beyond 3 Tesla, you can go to 7 Tesla, of course. This is my brain scanned by Todd Harrington, John Polimeni at the Martino Center. And you can see these COM system fibers that I talked about. This is a comparison of a dark field microscopy section by Eduardo Alio, where you can also see these COM system fibers here. Now, even beyond 7 Tesla, in vivo imaging is, of course, ex vivo imaging. And this is one scan that's remarkable. I'll talk about it more in this next slide. But if you just compare the two, there's so much more detail in the ex vivo scan. This scan was spearheaded, or the project led by Brian Edlow and the team at the Martino Center again. And they built a custom coil and they scanned a beautiful brain over a whole weekend. And that data set is openly available. It's amazing just to look at anatomy, just as a reference image. So be sure to download it and look it up. And it can now be used as a like optimal definition of anatomy of this region. So this is the integration of that scan into our software LeadTBS, where we can now look at electrode placement relative to such a reference scan with all these details in between these small structures. So atlasing atlas resources are yet another way to study the basal ganglia, because if we cannot image them as precisely in vivo, maybe at least we can use some atlas resources to define them. And I think the team by Beate Forstmann also did a great job in pioneering this approach. We also created one atlas that's part of LeadTBS together with Louis Collins and Mala Chakravarti from the MNI, which has histological sections, but also subthalamic nucleus subdivisions based on tractography and so on. So it tries to take the information that we have to best define the basal ganglia as a whole. And then once you have these atlas structures, how can you make use of them in the individual brain, right? We can, of course, normalize brains. Everybody knows that, and we did here. But we can now use a new software called Warp Drive to refine these normalizations at the regions that we're really interested in. So it's a bit like Photoshop for the brain. You can zoom in, you can say, I'm really interested in the subthalamic nucleus, and I want to slightly refine the ant's warp field by 2 millimeters to the left. And I use my mouse to nudge it in place. So this is the subthalamic nucleus now that you can see, and you can directly compare the outlines of an atlas that could be from histology, from your brain atlas, with your normalized patient's brain. And you can use the mouse, and then slightly nudge it, as you can see right now, on a warp field level. That could also be used to make the match between histological data and MRI data better, such as here a whole brain section of histology by Eduardo Alio, that we used Warp Drive to then manually refine the fit to the MNI brain with that. You can see that this is an MNI atlas, and this is the histological section. So we draw a line, and then Warp Field nudges it to the atlas. By doing that, we can create these playgrounds of high resolution resources that can be precisely matched to our patients' brains. And then we can use these in vivo, but also ex vivo atlas resources together to study the patients. Another option to define then white matter tracts in the basal ganglia region is histological mesh tractography, also introduced by Eduardo Alio. And he used that method on histology to basically study these very thin, small bundles between, let's say, the subthalamic nucleus and the pallidum. Of course, we can also use diffusion MRI, but we have to be really meticulous about it, a bit like Eric Mittelbrooks did here, to manually define and trace these a priori known tracts. That's yet another atlas that's openly available within LeadDBS. So you can use that to augment what you can see in the individual patient. And of course, we can use techniques like polarized light imaging, as we've seen this picture before. So in my view, the optimal conclusion is here to integrate and combine resources of the in vivo part from your specific brain of study with atlas data. And we need good techniques such as Warp Drive to fuse them together. Now, quickly, a bit about the application side of things. I mentioned I will talk about the brain stimulation quite a lot. So here, millimeters really matter. That's why it's the optimal example to highlight why all this precision is necessary. You can see 2D brain stimulation electrodes here that are just 2 millimeters apart from each other. This is an optimal stimulation site within that small structure of the subthalamic nucleus that I talked about. And the one that's closer to it led to 80% of improvement in clinical outcomes. The one that's further apart of it, only to 25%. And again, that's just 2 millimeters difference. So we need good imaging, and the best we have to resolve these differences in the first place. We used to define optimal tracts as well, a technique called DBS fiber filtering, where we basically can attribute fiber tract activations based on clinical outcomes. So we can filter which tracts were optimally engaged with the top responding patients. And we have done so to just name one example, in a larger cohort of 50 patients with obsessive compulsive disorder. We found out this tract, if you modulate it, patients get better. We made that tract openly available within LeadDBS, and within 6 months' time, we had like 4 studies that replicated the effects to some degree, right? So we can see that by now, with that precision imaging, we can certainly find models that can be replicated across the globe, actually. And I think as a final outlook option, one goal would be to transfer these precision neuroimaging concepts into the operation room. Here we see a new software, again by Simon Oxenford, where we can model where we are in the brain live and trace it to the big brain and to other atlas resources, and then to integrate it with electrophysiological data that we record from the patient. So, we put everything together. We have the great scanning, we have the ultra high field resolution, and then we have Warp Drive to put them together. And then we can use it in the operation room to really wiggle the millimeters back and forth. So as take homes, I think the basal ganglia are involved in most, if not all, we do. So they are important, probably also to you and to your science. They are challenging for neuroimaging. And I think a combination between in vivo and atlas resources is really fruitful going forward. Deep brain simulation can open a window into the brain to peek into what's happening down there in these obscure structures. And I think there's high future potential for clinical neuroscience. And again, also for all the imagers out there, there's a clinical need for high resolution data sets. So share your ultra high resolution data sets with us. They can be really helpful to study patients. So thank you very much. This is my team in Berlin. I will be moving to Boston this September, and our lab will be hiring there. So feel free to reach out, check out our lab website, netstim.org, or all these resources that I talked about most of them are part of LeadDBS software that you can find here. And there's also a podcast about stimulating brains online available that I curate. So thank you so much for your time. And of course, in the live sessions, we're more than happy to take all your questions. Thank you.